Yes, good evening everyone. Welcome to Lukmaan IAS. I hope that you are doing fine. In this particular Takkar perspective lecture series, what we are doing is we are providing in-depth analysis of today's important news, important current affairs relevant to our examination, both prelims as well as mains examination. Although the more focus is from the mains perspective, but yes, the same topic may be relevant to our prelims examination. So we are providing inputs for both mains and prelims examination. Like for example, today's topic, how gene therapy could cure cancer. Now we all have read about one of the 13-year-old teenager who have been cured is of cancer with the help of, we can say, base editing technology. One of the gene editing technology evolved in recent times. So such kind of topics we are understanding and they are relevant to our examination. So what you should do, this is half an hour session and you can make your own notes, one-page note of each and every issue, and that can be revised for the final examination revision or whatever the test series you are giving here. Okay, so yes, first of all, I will give you brief overview of this session. First of all, we will understand the context, why it is in news. Thereafter, about gene therapy, how the gene therapy in case of T-cell acute lymphocytic leukemia, which is also being called as TALL. Okay, then what do you mean by base editing? How gene therapy can help treat cancer? What is the significance of this particular gene therapy for cancer treatment or overall development at present and future? And finally, practice mains question and prelims question. Also, we will see the PYQs also from time to time. For example, when we are talking about this gene editing technology, we will also see the questions that are from biotechnology section. Okay, so this is explicitly mentioned biotechnology where in GS3 paper, science and technology syllabus. This is the relevance you should know. Okay, look at the previous year question. 2017 में एक क्वेश्चन ऐसे ही था, stem cell therapy is gaining popularity in India to treat wide variety of medical conditions including leukemia, thalassemia, damaged cornea and several burns. Describe briefly what stem cell therapy is and what advantages it has over the treatments, other treatments. Okay, so simply you should understand that such kind of therapy which are gaining popularity or in news, especially in medical science, the stem cell therapy पूछा है, तो of course gene therapy can be asked in the examination this year. So you should be knowing about the significance, especially for medical science. Okay, so that's why we are dealing with this topic. So the context is recently scientists in UK have applied their particular research on one patient, the teenager girl Alicia, who is having this. Okay, so T-cell acute lymphoblastic leukemia. Leukemia is, we can say, that scientific name for cancer. Okay, lymphoblastic is basically one of the variety of blood cancer. Okay, so it is one of the rare blood cancer, TALL. Okay, so T-cell acute lymphoblastic leukemia, and what happens in this particular type of cancer, I will explain that, but this treatment has been successful to some extent. Okay, although this is just a trial of this technology, which we are calling it as gene, let's say therapy, or in that we can say base editing technology. This can be asked in the prelims examination also. I will explain how in the past UPSC has asked question from this area. Okay. Yes, about gene therapy. Now, we all know that our genetic setup, or simply we can say genome, made up of number of genes. Okay, and these genes are nothing but a kind of, we can say, information, a coded information. Now, how this coded information is given in terms of bases. Okay, how many bases are there? 4 bases are there. Which are these 4 bases? You should know: A, G, C, T. Remember this. A is for adenine, guanine, G is for guanine, C is for cytosine, and T is for thymine. We learn this in biology 11 12 standard. If suppose some students are not having that subject, so you should know the basics. So these are nothing but a letter, and the combinations of letter gives a message or a word. Simply that is nothing but gene. Okay, that's why we generally say that genetic information. When we are talking about the combination of all these bases, simply we are talking about the genetic information given in the coded information in our, we can say, whole genome. Now what happens is, whenever such kind of gene is having some misarrangement of these, we can say that bases, that may result into some terminal disease, and one of the example is basically cancer. In this particular case, it is, we can say, blood cancer, lymphoblastic cancer, and that is acute, very, we can say, severe kind of blood cancer, which is observed in both children as well as adult, but yes, with the age this possibility goes on reducing. Now in gene therapy, what is done is, this information that is misarranged of these bases, it is edited. So what kind of editing is done? It can be fixed, it can be replaced, or it can be supplemented with the healthy gene so that it can function normally. Simply it is gene therapy. It is gene therapy. Okay, now we all know that yes, simply we can replace the damaged gene, we can inactivate the gene that is functioning incorrectly, introduce new gene into the body that may be needed for normal function. A variety of, let's say, permutations and combinations can be done. This is how gene therapy works. Now gene therapy in case of T cell leukemia. First of all, we should understand what are T cells and what is T cell leukemia. T cells are basically T lymphocytes. They are part of white blood cells. They are part of white blood cells. कहाँ पे तैयार होते हैं white blood cells? Bone marrow में. Okay, so bone marrow where this white blood cells or T cells are produced. Now what happens? Let's say first understand the function of T cells. We say basically they are, we can say that, fighter cells. White blood cells, we all know that they are fighter cells. So any foreign antigen suppose enters into the human body, simply they are alerting its immune system. They are alerting the body's immune system, and thereafter we can say some antibodies are produced in the body which can neutralize that antigen or foreign body. Okay, this is the function. Some of them become T memory cells, memory T cells. Memory T cells remember the information of that foreign object, and in suppose in future, suppose similar kind of foreign object organism enters into body, then they detect that and immediately start alerting its immune system. इसी तरीके से हम बोलते हैं कि कुछ वैक्सीन जो इनएक्टिवेटेड वैक्सीन होते हैं, such kind of vaccine, for example polio vaccine. So simply our body is getting aware about such kind of foreign microorganism which may in future enter into our body. So simply such kind of memory T cells are there in our body. This is the function. Now what is T cell leukemia? Leukemia is cancer, and cancer is basically the fast, uncontrolled, simply we can say uncontrolled growth of cells, and in this particular it is T cells. Now what happens in TOL, that is T cell acute lymphoblastic leukemia. Lymphoblastic means basically it is killing other T cells. Some of the T cells are killing other T cells and white blood cells. Okay, and because of that, what happens is it is resulting into weak immune system. It is resulting into weak immune system. So of course, because of that, what happens is the different organs are not working properly and the person may die. This is T cell leukemia. Now, gene therapy in case of T cell leukemia and this particular tall. What happened in this Alicia case? How the scientists worked out this particular case? Let's understand that. What they did is they first understood which particular genetic information is misarranged. On the basis of that, what they did is they identified the donor T cells from the healthy donor with similar kind of genetic information, and what they did is they did base editing. They used base editing technology. So समझो इंफॉर्मेशन में ऐसे कुछ होगा और ये मिस अरेंज होगा और ये G को A और T को C करना था, तो इन्होंने क्या किया, ये G को हटा के इनको रिप्लेस किया. This is called as base editing technology. This is called as base editing technology, and this is used here in this particular Alicia case, gene therapy. Okay, so let's first understand. Yes, I have explained you just now this particular acute. What is base editing? Base editing is new genome editing technology that enables direct or irreversible conversion of specific DNA base into another at a targeted genomic locus at the particular point, targeted point. So we all know, just now we have understood that there are 4 bases, and the combination of 4 bases result into genetic information or gene simply, and at particular location, if suppose that information is misarranged in our body, that may result into, we can say, lack of secretion of particular proteins, or it result into some kind of disorder, disease. So to treat that, such base editing can be done, and this technology is used in this particular case as a part of gene therapy. Understood? Okay. Now, how gene therapy can help treat cancer? Just now, let's say first of all, I will explain that what happened here in this particular case. From the healthy person or healthy donor, we can say, I will come back to this case instead of that. Okay, so healthy T cells were extracted from a donor and put through series of edits. The first base edit blocked the T cells targeting mechanism and removed the chemical marking called as CD. ठीक है, तो ये जो है वो second step है. They removed the chemical marking called CD7 which is on all T cells. Technical things है, आपको याद नहीं रखना है एग्जाम के लिए. But yes, what is important here, the process. What they have done, they have taken the healthy T cells from donor areas, donor, and then they have edited its T cells genome. ठीक है, or we can say genetic information that is required for that patient. And finally, let's say the third step is prevented the cells from being killed by the chemotherapy drug. This is also important. Okay, because if suppose chemotherapy is killing those donor cells, then what is use of that? So all these things were required, and thereafter the T cells were programmed to destroy all cells that is cancerous and protective with CD7 as marking. ठीक है, तो जो भी cells थी वो सारे उसको destroy कर देगा, and there can be now the second treatment where there will be healthy bone marrow transplant will be done. Healthy bone marrow transplant will be done, which will now boost the patient's immune system. इस तरीके से हमें ये चीज समझनी है. Understood? This is how it is going to deal with the or treat the cancers. Okay, so gene editing or base editing, now you have understood what is the significance of gene therapy at present and in future. You should know not only for cancer but for different genetic disorders. This is going to be helpful. Why? Because it is efficient. Why? Because efficient, we are saying that it is taking less time. Along with that, it is effective. It is resulting into, we can say, some kind of positive results. And thereafter, you should also understand that what we can say simply long term. At present, only the palliative care is given to the such kind of terminally ill patients, and at simply long term solution to such kind of medical problems can be we can say provided with the help of gene therapy. Okay, so number of advancements and breakthroughs are being made in gene therapy, and it will soon become clinically realistic approach in cancer treatment. So this is good thing. And in this particular gene editing technology, yes, in last 1 decade, CRISPR-Cas9 is in news. CRISPR-Cas9. This was asked in prelims examination. Let me show you the question. In 2020, chemistry Nobel Prize went to the female scientist had done research in the CRISPR-Cas9 टेक्नोलॉजी. ठीक है, तो उस टाइम पे भी काफी न्यूज में था. सो इन 2019 प्रीलिम्स एग्जामिनेशन, व्हाट इज Cas9 protein that is often mentioned in news? If suppose the students are aware about it, simply they can mark it correct. A molecular scissor used to target the in a targeted gene editing. So simply CRISPR-Cas9, how it works? CRISPR-Cas9. Okay, this it is acting, it is nothing but a kind of, we can say that, enzyme. It releases enzyme, it acts as a molecular scissor, and at a particular location of a gene which is to be edited. ठीक है, तो वो उसको edit कर देता है, acts as a scissor, molecular scissor. So you should know that there is release of enzyme, and it is directly, we can say, cutting that gene. It is related to gene editing technology. Remember this. Okay, and it is part of overall biotechnology. Now, what can be the question from the mains perspective? What do you mean by base editing? How does base editing help in curing cancerous T cells? So one is from basic half part, second is from current affairs largely. Okay, so yes, base editing, just now we have talked about base editing. There are 4 bases, a combination of which the overall genetic information, gene setup is there in all living organisms. 4 bases के नाम लिखने हैं आपको, एडेनाइन, ग्वानाइन, साइटोसाइन एंड थायमाइन. ठीक है, तो इनका जो भी कॉम्बिनेशन होता है, दैट इज, वी कैन से दैट जेनेटिक इंफॉर्मेशन, एंड मिसअरेंजमेंट ऑफ ऑल दिस रिजल्ट इनटू सम काइंड ऑफ डिसऑर्डर, डिसीज, एंड दैट नीड्स टू बी corrected. So correcting all these bases is nothing but base editing. ठीक है, and yes, in some of the cancers like this one, lymphoblastic cancer, leukemia, or we can say that the T cell acute lymphoblastic leukemia में कैसे treatment हुआ है, just now we have understood. Fine. Okay, so in this manner we should be preparing for biotechnology, and you should know that biotechnology से related जो recent developments है, हम उसको contemporary aspect बोलेंगे, last 3-4 साल contemporary developments. Be it 2017 में जो stem cells का पूछा था, stem cells therapy. If you are talking about, let's say, biotechnology, of course COVID-19 pandemic is very important. Whatever the technologies that were used for development of drugs, vaccines in COVID-19, that is very, very important for our examination. It is part of biotechnology, right? Okay, so yes, these are all, this is the discussion that we wanted to have. I hope that it has been fruitful and you make notes out of it, and that will be used for final revision before, writing the answers and even in the final examination. Okay, fine, thank you.